Okay. So for chapter 17, because I'm going to kind of skip through just giving you the essential parts on this of things. You also have a couple of, when we gave you those worksheets the last time, there was the ones that you couldn't do in there, something with the aliens on there where it had the DNA code and stuff. After today, you'll be able to do that worksheet. It had stuff for this in there. So last time we looked at the structure of the DNA, and the DNA is going to code for the instructions to make the proteins in there, and that's what we're going to look at. And RNA is your instructions for making protein, but it does it through an RNA intermediate. So you've got your DNA that you start out with, and you're going to go through a process called transcription in there where you're going to make an mRNA from it. And then that mRNA will be read through a process called translation to actually make the polypeptide, which ends up being your protein at the end. And so what is going to actually do the reading in there is the ribosome. So if you're in a prokaryotic cell, all of this just occurs in your cytoplasm because you just have a cytoplasm. You don't have a nucleus for any of it to occur in. If you're in a eukaryotic cell, you're going to have transcription occur in the nucleus, and then translation will occur in the cytoplasm. So they're in two different locations in the eukaryotic cell. So transcription is your synthesis of RNA that uses DNA as a template. So with your DNA, the way you copied it to make a new copy of the DNA when the cell splits, it's a very similar process that you're going to go through for transcription in there. And instead of filling in the base pairs to make another DNA, you're going to fill in the base pairs to make an RNA. The good thing is most of them are the same. The only one that is different is instead of where you would put in a T or thymine, you're going to put in U or uracil in place of it. It's still going to be the G and C pairing together. It will just be A and U pair together this time. Then translation is going to be where you synthesize the polypeptide from the mRNA, and the ribosome is going to be what reads it. So initially when you make your first RNA in there, before it's modified, we can refer to that as your pre-mRNA. Your DNA and that pre-RNA are going to have all kinds of other things in there that you don't really need. It has to get cleaned up basically to have just the instructions for making that gene in there. So with your genetic code, we know we have only 4 different nucleotides, the A, U, G, and C, but we've got 20 different amino acids. So you have to use a combination of 3 different nucleotides in there to code for 1 amino acid. If you just used 1, that would only allow for 4 different options in there. If you use 2, that would only allow for 16 different combinations. So that's why we have to do 3. And that's going to allow for 64 combinations, which means your genetic code is going to have some duplication in there because of that. So it's a triple code in there, and that is referred to as your codon, which is in here a little bit later. So you have your DNA template here that you use, and you'll notice that this strand here is complementary to this strand here. They would fit together in there. Their base pairs are going to match up. So it's going to make this one that is your mRNA. And each of the 3 letters on here form a codon, and that's what's going to code for a specific amino acid in there. So just like when you copy DNA, you've got your template strand in here that's going to provide the pattern that you use to make your RNA. So the codons are going to customarily be written from the 5' to 3' direction in there, and they're read in the 5' to 3' direction as well, just like it is with everything else. So 61 of those 64 codons are going to code for an amino acid. 3 of them are going to be stop codons that are not going to code for an amino acid. And that's how you know when you've reached the end of the line or the end of the instructions. When you go through and read it, you're going to know where to start reading because you start at the first AUG in there. So if you've got a whole sequence going along, say like going along here, you go along and you're looking at these 3-letter codes. You start where you find the first AUG. And that one is your start codon. So you just go along until you find that. That's how it knows when to start reading. If you start reading anywhere else, your reading frame is going to be off and you're not going to read the proper instructions. So it's kind of like where when you read a page, there's spaces in between the words. But if you squished all of those words together and there was no spaces in between them, and you go to read it, you're not going to be really sure what the reading frame is. And some of the words, the end of another word and the start of another word could combine to make a new word in there, and you're going to get the wrong message out of it. So that's why you always want to start at the first AUG on here. Those ones that I gave you last time, they're just for practice. So this genetic code is nearly universal in there. It's gonna, which is nice because it allows you to transplant genes from one species to another. There are a few little exceptions they've found in there with a few different codons other than the standard ones in certain unicellular eukaryotes or some of the genes in the organelles of some species. But for the most part, it's really close to universal in there. It's only been recently that they've even found anything that had a few exceptions in it. So this is your codon table here. And this is going to be how you know which amino acids it's going to code for. So with AUG, which usually gets pulled off at the beginning, it's the start, but a lot of times an enzyme is going to remove that amino acid in there. It's kind of like you don't need to have the wrapper on a pizza. You pull that off before you make a pizza. No matter what kind of pizza you make, you still take off the wrapper. So when you look AUG, if you look on here, the first letter you find it along this side here. So A is right here. The second letter you find across the top. And when you come together with those two, it's going to bring you to a box on there. And sometimes you can just look in the box and go, okay, yeah, all of the 4, that one is AUG. It's not too hard to pick it out of 4 choices. But the third letter is usually over here on the right, so that you can look and go, okay, the G is right here, and find where it is. So if you find letters, first letter, second letter, and third letter, and you have, it's where all those lines cross, is where you find the codon in there. So nobody memorizes the codon table in there. The codon tables are readily available. There might be a few of them. You end up, if you go through like what I do doing these examples, there's a few of them I have memorized just because it makes it easier to put it on the board if I know I put in a stop and I know I put in a stop at the end of it. But otherwise there would be no reason to memorize them. For the most part, this gets done computerized now anyway. So the next one, so this would be your code for methionine. So what would CCG code for off of that table? Proline. So is there anybody that doesn't see where we find that? So C over here is going to bring you down to this box, CCG, PRO. What about CGA? So arginine. And then last one, UGA. So that one is a stop. And normally in the DNA, you're going to have some additional codons that go beyond that in there. It means it's reached the end of it to stop reading it, and it no longer is going to add amino acids on. So what you have then for your amino acid in there is your gene. And remember, that's only about 1.5% of your DNA. So this is likely going to carry on way beyond there, and there's going to be a sequence way beyond the start in there too. So easy enough on how you read that to find out what the amino acids are. So when you read this, you're going to use an RNA polymerase is going to be the enzyme that is going to be involved in this. So where you copied the DNA, you used a DNA polymerase to add on. When you're making an RNA in here, you use an RNA polymerase. So at the start of it, we're going to have a regulatory sequence in there called a promoter in there. That's going to allow the RNA polymerase to attach and initiate the transcription process. Then at the end, you've got the stop sequence, but you also have a terminator sequence in there that is going to help with the stopping process and pulling it off. It's slightly different in prokaryotes and eukaryotes. Sometimes it's going to make just this long tail on there that is going to hang off there and create tension to help pull it off. And that's about the only purpose it serves in there. So the stretch of DNA that's transcribed into the RNA is the transcription unit. So for this class, as long as you know that it's an RNA polymerase, not a DNA polymerase that you use for this, that's good enough. But there are multiple different types of RNA polymerases. In eukaryotic cells, they know they have at least 3 types. There's just a single type in bacteria in there. So when you start this, you have the nucleotide where the RNA synthesis actually begins. You've got the promoter that helps to regulate it, and that's going to be several sequences ahead or upstream of the start in there. You've got these transcription factors that are going to help to mediate the binding of the RNA polymerase on there and initiate the transcription. It's one more thing that's going to help in regulating the process, because you do need to have all of the pieces of the puzzle to get what you want out of it. So the transcription initiation complex is going to be the transcription factors, the RNA polymerase, and it being bound to the promoter. In the promoter, there's this sequence of DNA that they call the Tata box on there. That is a whole bunch of T's and A's in there that are about 25 nucleotides upstream from the start. So it's one of the things where they found these patterns in there. And that's how a lot of these things get named. So you've got the transcription unit in here where you've got the promoter, the DNA in there. So it's going to open it up in here so it can read the DNA transcript in there. It's going to open up the DNA and make the RNA transcript, and then the DNA will close back up in there. And you've got this mRNA that you get to send out to the cytoplasm where you've got your ribosomes that can do the reading in there. So just like with the DNA where you had to untwist it in there to be able to read things, you're going to have to untwist it here as well. And you expose about 10 to 20 nucleotides at a time in there. Several genes can be transcribed simultaneously by several RNA polymerases. So you can have multiple genes being made into an mRNA off of a chromosome or a piece of DNA at any given time in there. So the RNA processing, when you've made this pre-RNA pre-mRNA in there, it's going to have the ends be altered on there. So some of the things that you have on there are a 5' cap in there, which is a modified form of guanine that gets added onto the 5' end after transcription. And there, a poly A tail in there can be added onto the 3' end. And what it adds on there is between 50 and 250 more adenines in there. So my example there just showed a few amino acids being added on. Typically you're looking at hundreds on there, hundreds of amino acids. It's just that we'd spend all day going through the example on the board, just coming up with the sequence in there. So to have 50 to 250 more adenines added on to the end of it really isn't making it that much bigger in there. So these things being on there seem to help facilitate the transport of the mRNA from the nucleus in there. It helps to protect it from being degraded in there, and it also helps the ribosomes in attaching. So we have RNA splicing that's going to go on. So you've got this big mRNA here that has come out. It's going to have the 5' cap, the poly A tail on there. And we've got regions on there that are called exons and introns. The exons are the expressed regions in there, and the introns are the intervening sequences. So these are just extra sequences in there that are not read in translation. So those have to get pulled out, and then you have your processed RNA here, that what you end up with is a much smaller piece. So there's all these little extra segments in there, because remember, what you read here is only 1.5% of the DNA. So we've got what they call SNRPs on there, which are short nucleotide sequences at the ends of each intron that's used as a signal for the splicing in there. So that's how it knows when it's reached a piece it needs to splice out in there. And the small nuclear RNA in a SNRP is going to have about 150 nucleotides in there. So you can have several SNRPs joined together with additional proteins, and that's going to make up a spliceosome in there, and that's going to help in the degrading and of the introns and then the rejoining of the exons to make the new piece in there. So this is showing it here. You've got this big intron in here, this section you need to get rid of. It folds it up in the spliceosome. You've got those sequences in there. That's going to allow these two ends to get glued together. You cut out this part and degrade it in there. So ribozymes are going to be RNA molecules that can act like enzymes in there. And RNA is able to do this because it's single-stranded in there, and it allows it to bind onto base pairs in there. So it can have a three-dimensional structure, which is part of what you need for an enzyme to have catalytic functions in there. It can have functional groups that can participate in the catalytic functions in there. And it's able to hydrogen bond in there to help with the catalytic functions. So remember, a lot of times what enzymes do is they might squeeze something so that the pieces are closer together in there and the interaction can actually take place. And the RNA is able to do that too because of those functional groups and the extra hydrogen bonds that it can do. So these introns in there, at first they didn't really understand what they did. And that's part of the reason why initially they figured humans had 100,000 different genes. But because of these introns in there, they're figuring it went down to 30,000, and now they're thinking we're down to maybe 10,000 genes in there. What it does is it allows you to do this alternative splicing in there, where you can recombine those little exons in different ways. So you can have one gene that's got multiple exons, And if you recombine different exons in there, you can get several different amino acid combinations out of it, or different proteins in there. So we refer to that as exon shuffling in there. So it's kind of like having crossing over within the DNA itself in there to make new combinations in there. So say you were to have a gene, or you've got your piece of DNA here, and you've got a gene sequence here, or an exon here, and another exon. So we've got A, B, C, and D. Depending on how you splice that, you could have a gene made from it that's going to use like A, B, and C, A, B, and D, B, C, and D, B, and D, A, and D. So you can see how one gene can end up with multiple combinations, and this isn't all of them that you could get from there. So that's why they overestimated how many genes we have, is because they didn't realize that we could recombine that. And that's why you can have one gene code for so many different things in there. So when you're going along reading your mRNA in there, you've got a substance called tRNA in there that's your transfer RNA. It's made up of RNA, but what it is it's going to go along and it's going to match up and pair up your amino acid with the codon. So the tRNA is actually this folded piece of RNA here, and it's going to have your amino acid attached to one end. And at the other end, you've got this region called the anticodon, which is going to be complementary to the codon. So if you've got a codon here that is CCG, you're going to have an anticodon that is GGC, that is part of your tRNA. And you're going to have your amino acid at the other end of it. So by having that anticodon on there, it's a way of verifying when it's read it that yes, we do have the right amino acid here. It's kind of a backup, like a spell checker on there to say, okay, yep, brought the right amino acid here in the instructions. So the enzyme that's going to carry this out is the amino acyl tRNA synthase in there. So when you look at the codon table, I'll just scroll back to it since you probably don't have one in front of you. What you'll notice is a lot of times you've got this whole block of them, and the only thing that's different is the fourth or the third base pair in there for all four of those. Otherwise, the first two letters are the same in there. And that's not just random that that happens in there. It contributes to what we call a wobble. In there. So sometimes it will be a little bit flexible with what it will allow to pair at the third spot. It may not require it to be an exact match in there. It's going to be more lenient and say, you know what, this anticodon here, this one is close enough, 2 of the 3 match, we'll take it in there. And it's still going to make the same protein in the end though, because of that duplication that you have in the codon table in there. So part of what makes up your ribosome is going to be some rRNA in there that's going to help with its structure in there. That's what rRNA is, just part of the structure of the ribosome itself in there. So in bacteria and eukaryotes, you're going to have different types of ribosomes. They're slightly smaller in the prokaryotes than they are in the eukaryotes in there. They measure them in what they call Speedbergs in there. So one is in 80s for 80 Speedbergs in the eukaryotes, it's 70s in the prokaryotes in there. The different types of ribosomes is actually really significant for certain drugs that you would take, because if you're going to take an antibiotic in there, you want that to kill the invader. You don't want it to kill your own cells. And if you target it to attack a 70s ribosome, which is only in prokaryotes, it's going to do far less damage to a eukaryotic cell in there, because what it's destroying is the 70s ribosome. It will still do a little bit of damage because you have some of the, you've got bacterial type ribosomes that are in your mitochondria in there. Because of those with the endosymbiont theory that they were engulfed, they still kept the prokaryotic ribosomes. But the rest of your cell is going to have the eukaryotic style ribosomes in there. So a couple of drugs that will do that are tetracycline and streptomycin, and they're common antibiotics that will attack the 70s ribosome. So the ribosome is going to have 3 different sites on there for tRNAs. So when it comes along, it's got a P site, an A site, and an E site in there. So you've got your 2 subunits of it. They're going to attach onto the mRNA. And it will allow 3 different tRNAs with 3 different amino acids to come in there at once. The P site is going to be the one that carries the growing peptide in there, where you're going to add a new peptide on there. You're going to have the A site in there that's going to, that's the one right here where it's going to be added onto. The A site is in front of it. That's where you're going to have the new accepting the tRNA in there. It will accept a new tRNA to come in that's going to get eventually moved to this site. And then the E site is the exit site in there, where you're going to have the discharged tRNA leave. So the tRNA is going to drop off its amino acid, and then it's going to head back out into the cytoplasm to be recycled. So the next time you eat some protein and you break it down into amino acids, you can hook it up to those tRNAs again. So this is showing here when you build it. You've got this new tRNA coming in with an amino acid. It's going to get added on in there. They're going to move down, and then it exits out the E site in there. So it just comes along, and as it moves from the A site to the P site to the E site to gone, to be recycled. So we want to be able to make sure we regulate this process, and the more steps you put in it, the more options you have for regulating things in there. So the initiation factors are going to be proteins that are going to be required to bring the initiation complex together in there. And it's always going to be synthesized from the N terminus to the C terminus, which means when you're looking at an amino acid, if you remember, you had the amine group and the R group. This is what they mean by the N terminus and the C terminus in there. So the elongation factors are going to be involved in adding on the amino acids to the previous one at the C terminus. So just like you do 5' to 3' on the DNA, there is a certain direction you move and add on to the amino acids in there. And then you've got release factor in there, which are going to be protein shapes that are going to bind to the stop codon in the A site. And it's going to cause it to add water instead of an amino acid, which will end up hydrolyzing the bond so that you can have it release in there. So one of the things that can happen, if you need to make a lot of protein in the cell, this system actually really allows for a lot of amplification of that. You can use a DNA to make multiple sets of the instructions, so multiple mRNAs. And then when you have that set of instructions, you can have multiple ribosomes reading the instructions at the same time. So that's when they talk about polyribosome in there, is having multiple ribosomes translate at the same time. So you've got one here that's reading, one here, one here, one here, that's finished and released in there. So it just moves along the assembly line in there, and when one finishes at the end of the assembly line, it makes room for another one to come in. So you can actually make a huge amount of protein that way. So this is showing multiple ribosomes reading along an mRNA on an electron microscope picture in there. So after you've made this chain of protein in there, now you need to be able to get it off to where it needs to go. So it's going to go through some post-translational modification in there. We know that they're automatically going to fold up into their primary, secondary, tertiary, and quaternary structures in there, but we do a little bit more modification to it in there as well. So single peptides, or the signal peptides in there, are going to put marks on there that are going to destine it to go to the endomembrane system. So it's kind of like putting an address label on there. When you have it all done, this is where it goes, and that's going to help get it there. So the SRP is going to recognize the signal peptide, and it's going to help to escort to bring the ribosome to a receptor protein in the endoplasmic reticulum. So some things that can go wrong are mutations in there. If you are going to change the genetic information in there, it's going to change the message and the instructions on how to build that protein, and you're going to get a different product out of there. So small scale mutations in there, or a lot of times your point mutations in there, where you just replace one nucleotide in there with a different one. So if you replace a different nucleotide in there, what's going to happen? So there's three things that can happen. So being that there is the duplication in the code, you could replace it with the same amino acid in there. If you went from like CCG to CCC, both are going to code for proline. Those are probably the best case scenario in there because it's going to make the same thing. If you put in one that turns, say, instead of it going from CCG to now it's ACG, you're going to get a completely different amino acid in there. So it's going to make a different protein. And it can also turn it into a stop codon, in which case it's going to terminate reading it altogether in there. So the silent mutation is the one that has no observable effect on the phenotype in there. That's the one where it's the CCG to CCC. It puts in the same amino acid. A missense is going to be one where it switches out one of the amino acids, and a nonsense is going to cause it to stop. Ones that can cause a little bit bigger problem are the insertion and deletions. Sometimes an extra base pair gets added in there or taken away. And what that's going to do is it's going to switch the frame shift of everything else down the line. So that's again like when you squish all the words together and you have no spaces in there. You don't really know where you start and stop reading for each individual word, and it's going to make something completely different in there. So usually those are big enough that it's going to make something that's non-functional in there. So this is going to show some of them in here where you have a base pair substitution in there, an insertion or deletion in there, where you end up with the different proteins being made. So substances that cause this to happen are called mutagens in there. They can be physical or chemical agents that will do this. You can have spontaneous mutations where you're just going along and during that replication process you make a mistake. So it's going along very quickly, and for the speed that it goes, it's estimated that 1 in 10 to the 10th nucleotides are altered and changed, and that gets passed on to the next generation. So for the speed it goes, it is very accurate in there, but occasionally you get mistakes in there. Other things can contribute to mutations. X-rays will cause mutations, UV radiation, lots of different chemicals in there. Some of them are considered carcinogens because they cause mutations that are known to lead to cancer in there. So we talked about how you can have a gene for cancer but not necessarily get cancer. That mutation is one of the ways that you could have a mutation in there where that gene is normally turned off, and now all of a sudden you have a mutation in there that you make a start codon on there and allows it to be turned on. So let's see. When we look at the gene expression in prokaryotes and eukaryotes, the archaebacteria use an RNA polymerase that's more like the eukaryotic ones in there. With the prokaryotes, they can start doing translation before they finish transcription because it's all occurring in one space. Eukaryotes have to finish transcription and send the mRNA out to the cytoplasm before translation can occur. It's just because they're in separate locations that you've got the time difference in there. So that is the end of 17.